Hello dear audience. My name is Mert Kurt, and I would like to explain the basics of confocal laser scanning microscopy. So let's get started with uh, slides include. I have three main types actually: the introduction, working principle, and advantages and limitations of this confocal laser scanning microscopy system. I would have to move on the introduction part, and this is the important part, I think. So, confocal microscopy is a fluorescence imaging technique that produces exquisitely sharp optical sections through biological specimens by exciting a laser over the sample and collecting emission data point by point to reconstruct the final image. This is a 3D reconstructed structure, as you see. And confocal microscopy is used in clinical and research laboratories and is especially useful in studying live cells. Actually, it has three types. These are point confocal microscope and slit confocal and spinning disc confocal. This is known as NIPCO. So, choice will depend on application. Laser scanning scopes have high resolution and slow imaging frame rates, and spinning discs have high frame rates, which is advantageous in live cell imaging. When selecting a system, consider the ease of training users, the ability to adapt the system to future needs, and the expense to maintain and repair the system. So let's move on the comparison between confocal and wide field microscopy techniques. As you see, wide field image has blurry image, and confocal microscopy is so pure and resolution is higher. As you see in this photograph, wide field technique and or non-confocal technique has a scattered light, but confocal microscopy's laser light has point-like intensity. So this is too important in imaging, as you see in this picture. And what is the history of confocal microscopy? Marvin Minsky invented the confocal microscopy in 1957. So the technique essentially scans an object point by point using a focused laser beam to allow for a 3D reconstruction, as I said. Let's move on to instrumental design. So, yes, as we see in this picture, the CLSM works as by passing a laser beam through a light source aperture which is then focused by an objective lens into a small area on the surface of your sample, and an image is built up pixel by pixel by collecting the emitted photons from the fluorophores in the sample. If we have a camera in this place, we have an image like this with different depths in the sample. Assume that red particle is a cell, different cell, and blue one is a different cell, but they have different depth scales, but we see it on the camera like this picture. Let's move on to CLSM components, a breakdown of the system. The laser is line that can be chosen via selection device and are matched with the fluorophores used in experiment. Beam splitter, as we know all of the optical diagrams, this is a filter that separates the excitation from the emitted light in the fluorescence beam path of the microscope. Scanner, this is a unit based on two or more mirrors which guide the focused laser beam across the specimen, pixel by pixel and line by line. So objective lens, these are the heart of the microscope and mainly determine the optical image formation and resolution of the system. Z-control, this allows you to focus on any focal plane within your sample, and motorized Z-stepper allows movement in the axial direction in small step sizes, bigger than 10 nanometers, with high precision. And the most important part is pinhole. This is an adjustable iris in the intermediate image plane. It allows exclusion of most of the out of focus light from the acquired image and thus provides optical sectioning capacity. It defines the thickness of the optical slice and is dependent on the properties of the objective lens. The pinhole size can be set via the software on your computer, and the best trade-off between the efficiency of the light collection and optical sectioning is if it is set to 1 Ary unit. The unit is Ary units. So photomultiplier tube is a detector which detects the intensity of the light, and this has to be a highly sensitive detector. So we can see the parts of the confocal microscope, confocal laser confocal microscope on the diagram. This is the laser, and this is scanner. This is objective lens. This is our sample. And this is dichroic beam splitter, and this is the pinhole, and this is our detector, photomultiplier tube. So let's move on to principle. As you see, this is the scan control. We can choose it by it depends on the usage. So Galvo scanning mirror is seen on this photographs. You can move the laser beam on the XY plane marking field, as you see. The Galvo scanning mirror, if you make angles on a mirror, you can move the light. So this principle is same. So if you see this Nipkow disc has micrometric holes on it, and it turns. So let's move on the working mechanisms in 3 states. As I mentioned, in confocal microscopy there is no lamp, we have laser beam, and we don't capture an image, we get the intensity of light and we reproduce an image by detecting the light with photomultiplier tubes. So I I have to give some more detailed information about the XY axis movements. So one mirror tilts the beam in the X direction, the other in the Y direction. Together, they tilt the beam in a raster fashion. The beam is then brought to the back focal plane of the objective lens, which focuses it onto your sample. If your sample is fluorescent, part of the light will pass back into the objective lens. This light travels backward through the same path that the laser travels. The effect of the scanning mirrors on this light is to produce a spot of light that is not scanning, but standing still. So this is the second part, second stage. This light then passes through a semi-transparent mirror which reflects it away from the laser and towards the detection system. The first object in the detection system is the pinhole aperture, which is the intermediate image plane of the microscope. This allows only a small central portion of the light through the light detectors. So the last part, any light that emerges from the CLSM optical system may have a very low intensity, and so the photomultiplier tube is used to detect and amplify this light signal. And all of these complicated steps occur so fast that the display seems like it is showing a real-time image of the sample. Let's move on the applications and usage on the area. Actually, it's used in biological sciences. Yes. And quantum optics and nanocrystal imaging and spectroscopy. As you see, biological and medical sciences. As I read, there is a usage on encrypted data and optics and crystallography. So the advantage of confocal laser scanning microscopy has higher resolution than the wide field microscopy and imaging of thick specimen by optical sectioning and elimination of autofluorescence from outside the focal plane by using spatial filtering. Control of spatial depth by optical slicing. So even so it has advantages, the confocal system has limitations too. So it has drawbacks. What are the drawbacks? Resolution. It has inherent resolution limitation due to diffraction. Maximum best lateral resolution of confocal microscopy is typically about 200 nanometer. So the pinhole size. Strength of optical sectioning depends on the size of the pinhole and the intensity of the incident light. And if we use the fluorophores should tag the correct part of the specimen, and fluorophore should be sensitive enough for the given excitation wavelength, and it should not significantly alter the dynamics of the organism in the living specimen. And we have a photobleaching effect. Photochemical alterations of a dye or a fluorophore molecule such that it permanently is unable to fluoresce. So I have references also. Special thanks to our Professor Hüseyin Kurtuldu, and I would like to thank the listeners. So this is the end of my presentation. Thank you all.